For the next 3 hours, I'll be providing an overview of host defenses against infection. My name is Keith Krolik. I'm in the Department of Microbiology, Immunology, and Molecular Genetics. And as you can see, my email address is indicated at the bottom of this first slide. Feel free to use that email address if you have any questions about the material or anything else having to do with this presentation. The 3-hour presentation is going to be divided into 3 major parts, provided to you in 3 different files, in fact. The first part is going to concern areas of host defenses that we refer to as innate immunity. Innate meaning already there. It's not, there's no requirement for activating it. It's just there, it's innate. And so we'll spend the first hour together talking about innate immunity, but there are a few words that I'll say about other areas of host defenses as well as we go through the next few slides. The goal of host defenses in general is to use a diverse array of strategies to defeat an equally diverse army of infectious microorganisms. This is an amazing thing, the way we have evolved so many different tools and weapons to deal with so many different kinds of microbial pathogens that are out to get us. It just, it boggles the mind. When I think about all of this, I think about it in terms of layers of defense. We have the pathogen, the microbial pathogen, with the potential for really doing harm to us. And then we have the host down at the bottom of this schematic. The pathogen has to get through these layers of host defense in order to reach the host. And each of those layers provides a different kind of activity that will interfere with the survival of the pathogen in its quest to make its way to the host. The first layer of defense is maybe the most obvious and the one that we give least attention to when we're thinking about how we defend ourselves from infection. They're physical barriers. They're just basic walls of tissue that situate themselves between us and the potential pathogen. Skin is the most important, probably, well, maybe not the most important, but maybe the most obvious of those barriers because it covers the vast majority of the surface area of our bodies. As long as skin remains intact, it's very difficult for any average pathogenic microorganism to enter the inner workings of our body. Now, there are issues with organisms that can crawl their way into follicles and so forth, and we end up with cases of folliculitis and so forth. But the fact is that most of those sorts of infections are not life-threatening, and the skin provides us with a really nice barrier to infection. Now, if that barrier is breached, if the skin is broken by cut, abrasion, or any other kind of physical trauma, then potential pathogens can enter the body. And you might not think about that all that much, but if you were to ever walk through the average burn ward of a hospital, you could see right away just how important it is that skin remain intact to prevent various kinds of infection. The second physical barrier is provided to us by layers of epithelium that we call the mucous membranes. These mucous linings are sort of like walls that surround the entranceways into our body. So entranceways like the respiratory tract, or the genitourinary tract, or the gastrointestinal tract, and so forth, all have layers of epithelium that discourages microorganisms from moving laterally through the body. So even if you were to ingest or inhale a pathogen, the pathogen will pretty much follow the pathway, the hallway, down into those tissues and will be prevented from moving sideways to get to other areas of the body. And that's a really important feature of these physical barriers to infection. The other thing that mucous membranes do is they produce mucin, or mucus, converted to mucus. And this is a thick, viscous, liquidy sort of substance that moves across the epithelial layers and flushes any microorganisms that are trying to penetrate the mucous membranes, flushes them towards either the beginning of the hallway, the pathway, or the end of the pathway. In either event, it keeps those pathogens from adhering or sticking to host tissue and allowing them to set up shop to do us damage. So that, in a sense, is another physical capability that we have of preventing pathogens from gaining a foothold in our body. So we have skin, and we have mucous membranes with its secreted mucus that provide us with these physical barriers. Now, if the pathogen figures out a way to get through the physical barriers, or there's some damage done to the physical barriers, then we get to the next layer of host defense that I consider to be physiological barriers. These are barriers to infection that are they're not physically preventing microorganisms from moving anywhere. What they do is they prevent those microorganisms from thriving in their growth environment wherever they end up. So every pathogen has certain characteristic needs in order to be able to grow and thrive and do the things that they do. Certain temperature, for instance, certain amount of oxygen, a certain supply of nutrients, a certain type of surface area to attach to. There's all kinds of things that microorganisms need to be able to do in order to thrive in a particular environment. I brought with me three possible examples of these kinds of physiological barriers: fever, nutrient supply, and oxygen requirements. So first, with fever, the fever response is kind of interesting. Most microbial pathogens require normal body temperature to thrive, to really grow at their best. That's 37 degrees centigrade or 98.6 Fahrenheit. Just an elevation of 1 degree even in body temperature will prevent many pathogens from growing optimally and may even kill them. So fever responses are a very important physiological barrier to infection. Now, we didn't always realize this. And I remember back when I was a kid, my mom, who grew up during the Great Depression, had been exposed to or experienced lots and lots and lots of stories and even some physical personal experiences leading her to believe that fever was evil. Fever killed people. And in fact, back then, fever could kill people. Nobody could really control the infections that were causing the fever very well. And so the fever would last for a long time and reach very high levels, become very damaging and even life-threatening. So every time I ever got a fever as a child, even though there were antibiotics by then and everything was a lot better understood, it didn't matter to my mom. The moment my temperature went up, over 100, she'd be throwing me into a bathtub full of ice water. Some really fond memories of my childhood, I might say. Not really. Anyway, so I love my mom. She was doing the best she could, but fever, if she had been told that fever could actually provide some benefit to defending ourselves from infection, maybe she would have put me in water that was a little less icy. The second physiological barrier to infection has to do with nutrient supply. And there are a couple of different ways that nutrients can influence the course of an infection and the survival of an infectious microorganism. First of all, there are certain nutrients that certain pathogens require in order to grow at all. It's interesting that there are quite a few pathogens that require a really solid supply of iron, for instance. So being in the blood or in an organ that's really blood saturated is really good for them. And there are other nutrients as well, certain sugars and various other nutrients that certain pathogens crave that they can only find under certain circumstances, and if they enter the body in certain places and so forth. So that's one way that nutrients can influence the course of an infection. The other way that nutrients can alter an infectious progression is a little more sort of gross. The fact is that our bodies are just saturated with different kinds. Now, they don't do us any harm. They're often referred to as commensal organisms. Sometimes they're referred to as the native flora or the natural flora. They often not only are harmless, but are helpful. If you think about what it takes to digest food, for instance, we wouldn't be able to do it very well if we didn't have the help of lots and lots and lots of microorganisms that fill up the colon and help us digest our food. So we have lots of bugs living with us, and they're our friends. Well, they're also able to provide us with a barrier to infection by potential pathogens. If a potential pathogen were to enter the gut, just a few, you just happen to eat, that kind of greenish looking, enchilada that you have sitting in the back of the refrigerator. It's been sitting there for 2 weeks, and there's probably stuff growing in it. And if a little bit of that gets into your digestive tract, and there's certain pathogens, maybe E. coli, salmonella, whatever, that might be associated now with that kind of aging enchilada, those microorganisms, those pathogenic microorganisms, are going to go down into your gut and want to set up shop. But what they're going to be confronted with is a bazillion bugs, bacteria of all different kinds that are part of our normal flora, our natural flora. And they're all competing for the exact same nutrients that the pathogenic microorganisms are competing for. And so if you have a bazillion, commensal organisms, a bazillion is a big number, in case you didn't know, a bazillion commensal microorganisms competing with a few pathogenic microorganisms, very often the pathogenic microorganisms are just going to lose, and they may never be able to set up any kind of a niche in your gut, or if they do, it's for a very brief period of time, and then they're run out of town by the commensal organisms. So that's another way that physiological barriers can defend us from infection. The last of these characteristic physiologically dependent activities or scenarios can help us out has to do with oxygen tension in the body. If you look at different tissues in the body, you'll find that there's a different level of oxygen in many different places, many different organs. For instance, in the lungs, there may be the highest level of free oxygen available because there's so much oxygen exchange going on in the lungs. If a potential pathogen is in the lungs and once needs oxygen, it's going to have plenty of oxygen to keep it happy. On the other hand, there are other deeper tissues where, of course, there's always oxygen everywhere to a certain extent, but there are certain tissues, deeper tissues, where the level of oxygen is significantly lower. So if an organism were to really not just crave oxygen, but require oxygen in order to thrive, an organism that comes to my mind is pneumococcus or streptococcus pneumoniae. One of the major players in one form of bacterial pneumonia. Pneumococcus is what we consider to be a strict aerobic microorganism. Aerobic microorganism, that is an air or oxygen requiring microorganism. So this strict aerobic microorganism, if it were to be inhaled into the lungs where there's lots and lots of oxygen exchange going on, it is going to be a very happy camper and will begin to grow like crazy, will thrive, and eventually could develop into a bacterial pneumonia. And of course, pneumococcus is a highly treatable microorganism with antibiotics and so forth, but still it could be of potential life threatening sort of infection. There's other kinds of organisms, though, that are just kind of the opposite. They're considered to be anaerobic microorganisms, anaerobic. They may even be strict anaerobic microorganisms, some which find oxygen to be toxic. And if they're in a high oxygen environment, they may even be killed by that oxygen. anaerobic microorganisms. The organism that comes to my mind that's strictly anaerobic is the Clostridia, various kinds of Clostridia. One in particular, Clostridium perfringens, for instance, looks for places in your body where the oxygen tension is very low. So if you were to take Clostridium perfringens and put it on the head of a pin or something and jab it down into your thigh, deep down where the oxygen tension tends to be a bit on the low side, after a very short period of time, that organism is going to thrive and develop into a major case of gas gangrene. So it really looks for places that doesn't have a lot of oxygen around. Now, on the other hand, if you were to take a fistful of Clostridia perfringens and suck it into your lungs, it would go down into your lungs where there's all this oxygen and probably would do you no harm at all. Well, unless you choked on it, a fistful of Clostridium is a lot of Clostridium perfringens. Anyway, if you were to put it into an oxygen-rich environment, it's not likely to be able to cause disease. So what I'm getting at is that depending on where a microorganism, a pathogenic microorganism ends up, what route it takes in the infection that it's creating and so forth, it may or may not find the oxygen levels to be suitable for its own growth and survival. So here we have fever responses, nutrient requirements, and oxygen requirements that all can be considered to be physiological barriers to infection. Now, if the physical barriers are in some way breached, if we're cut or there's an abrasion or some physical trauma to these outer walls, and a microorganism gets in that's capable of adapting or mutating to the environment that it finds itself in and is able to get past the physiological barriers, then the next thing that we have going for us is this third layer of host defense that we refer to as inflammation, the inflammatory response. Now, the inflammatory response is a bit more sophisticated than these other forms of innate defense. There are actually specific cell types that are involved in specific functions that they perform. The reason that they're included in the innate immune system is that they're always there. They're always roaming the body, looking around to make sure that nothing bad is happening. And so this inflammatory response that we're going to be discussing in just a bit comes from particular subsets of leukocytes called granulocytes or macrophages. And as I said, we'll be talking about that in more detail in just a minute. Finally, if this pathogen were to breach the physical barriers, adapt to the physiological barriers, and maybe outrun the not quite so sophisticated leukocytes of the inflammatory response, we have one last chance to really get rid of them, and that has to do with what we refer to as adaptive immunity. Adaptive immunity is given to us mainly by lymphocytes, several different kinds of lymphocytes. We'll be talking about this in great detail in further discussions. So if all is perfect for the pathogen and it gets through all four layers of host defense and reaches the host, then the host is in trouble. And so there is a co-evolutionary thing going on constantly where the pathogen is figuring out ways to get through the barriers. And then we evolve so as to make the barriers more effective. And then the pathogen figures out how to get past those changes. And then we change and they change. So we call it co-evolution. And it's sort of a race right now to see who's going to be the ultimate winner in all of this, and who's going to survive and be the final species, that exists. Some days I'm just not quite sure who that might be. Anyway, so we have all these barriers to infection, and we're going to spend the next little while talking about barriers provided by these inflammatory activities that we were referring to before. Okay, so the main player in inflammation, as I said, are leukocytes. These are the white blood cells that travel through the bloodstream throughout the body, searching for signs of microbial invasion by constantly sampling the environment. So they're always roaming around looking for trouble. And when they're not roaming, they find themselves living in lymph nodes mainly and other lymphoid tissues. And we'll talk about lymph nodes later on. But lymph nodes are where a lot of these leukocytes will kind of move through looking for something, something to fight with. And one day I came across this GIF that's shown here, this little frog, and I looked at it for a second and I said, you know what? That is a leukocyte. That lily pad is a lymph node. And that fly that's flying by is something that the frog wants to get rid of. So that's got to be some infectious microorganism, right? So I saw this, I thought, I'm going to use this as an example of how these white blood cells are constantly searching for signs of microbial invasion. Also, I thought it was one of the cutest little things I've ever seen. And if you don't think it's cute too, then I don't know, maybe you ought to just delete this file and go home, because there's no hope. No. Anyway, I find it to be very cute. All right, so this is our definition of inflammation. At least it's the beginning of a definition. And we can go on to say that when signs of invasion and damage are detected, leukocytes will leave the bloodstream, where they're moving around most of the time, and go to the site of the infection. So this is a very defined process. People have been studying this for many years, and it's a really important thing because all these leukocytes are roaming around the body in the blood. That's great, as long as the infections that we get are only in the blood. But that's not usually the case, or it's not always the case. And sometimes we need to get those leukocytes out of the bloodstream and get them to the site of infection. And we call that process overall leukocyte recruitment. And I'm going to have a few words to say about it, but realize that this is a huge field of investigation, and there are just so many really cool things that people have discovered about how leukocytes can sense the presence of an infection and then go get it, you know. So one thing that we have noticed, or was noticed very clearly in the beginning of all of these investigations, is that leukocyte recruitment is associated with increased vascular permeability and movement of leukocytes from within the bloodstream to sites of infection, like I mentioned a second ago. By the way, anytime I put words in these slides that are something other than white, like this is increased vascular permeability shown in blue. Sometimes it's red, sometimes it's yellow. It depends on my mood. But anyway, when you see something like that, it means I think it's a really important phrase. So obviously you should think it's very important too, right? Okay. Okay, associated with increased vascular permeability. Vascular permeability is just, you know, how easily fluids and cells can get out of the vasculature, out of the bloodstream. So what happens during inflammatory responses, is that there is an increased amount of vascular permeability, which is what we want in order to get these leukocytes from the bloodstream to the site of an infection. It's got to get through the walls of these blood vessels. Here's a blood vessel. And if you look at this image, what you'll see is that there's like this thick white, here I'll use my pointer, this thick white wall. This is actually endothelium. This is the wall of a blood vessel. One of the things about blood vessel walls that wasn't obvious to me until, I don't know, I got a little bit older, a little more educated, was that blood vessels are not long stretches of tubes like garden hoses. That's the way I visualized it when I was a kid. In fact, blood vessels are made up of many, 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 many cells, endothelial cells, that kind of lay down. They're kind of elongated cells. They lay down head to tail, 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 and create this channel, this vessel. So there is contact between the endothelial cells, one to the next, to the next, to the next, and a little tiny junction in between, a gap. And they call it a gap junction, or sometimes a tight junction. And so in between these endothelial cells is this little tiny gap that allows fluids to come out pretty easily, not too easily. You don't want the blood just falling out of the blood vessels, but really doesn't let cells through whatsoever. It's too small. So what we're going to need to do is we're going to need to widen that gap and sort of open a door that allows the cells that you can see flying through the bloodstream right here inside the blood vessel. We need to get these white blood cells, these leukocytes, out of the blood vessel and to wherever the site of the infection is. So let's pretend for a second that the infection is going on over here to the right. And these leukocytes are sensing something is going on outside of the vessel. Now, the something has to do with products that are often made by the tissues over here to the left. I'm sorry, to the right. Did I say left? Right, over to the right, that are attractive to the leukocytes. We call them chemoattractants. Those chemoattractants are usually very small molecular weight molecules that can sort of ooze out of the damaged tissue, make their way to the blood vessel, might be a capillary or a slightly larger vessel, and just diffuse into the vessel, maybe through the gap junctions. In any event, they get into the blood vessel. They are designed to stop the movement of leukocytes and attract them to the source of the chemoattractant. So the leukocytes will begin looking for the source of the chemoattractants. Now, if they're inside the blood vessel, the first thing that's going to happen is they're going to kind of just sort of bash into the wall, trying to get over to where the infection is. So here they are over here. They're kind of sliding over to the right, and they're bumping into the inner face of the endothelial barrier that makes up the blood vessel wall. And at first, they just sort of skid along because they're moving at a pretty high rate of speed. They look kind of skid along the wall, and then there are these molecules called adherence molecules that are designed to grab onto certain surface molecules on the leukocyte. So we have adherence molecules or adhesion molecules, sometimes they're called adhesion molecules, on the inner wall of the blood vessel, and adhesion molecules that are on the surface of the leukocytes that attract each other, that bind to each other. And as they do, the endothelial cells of the blood vessel sort of grab onto the leukocytes by way of their adhesion molecules. And at first, there isn't much of an effect observed, but then little by little, the leukocytes get slowed down and slowed down and slowed down until finally they sort of are starting to come to a stop. And during the last few seconds while they're coming to a stop, they literally roll along the inner wall of the blood vessel. Some people call it rolling adhesion. And once they roll to a point where they find one of those gap junctions, which are now widened because of the increased vascular permeability created by other mediators that are produced by the damaged tissue, once they get to that gap junction, they're able to wiggle their way between two endothelial cells and then ultimately reach the infection that's going on over here to the right. So these chemoattractants from infected or otherwise injured tissues attract them to this inner wall, and then the increased vascular permeability that's stimulated or activated by certain mediators coming from the damaged tissue allow the leukocytes to actually leave the blood vessel. One thing about these chemoattractants, there are several different kinds. We'll mention another major kind in just a little while, but one collection, it's a variety actually of molecules referred to as chemokines. These chemokines are very potent chemoattractants that come from the damaged tissue. Okay, chemokines. All right, so here we have that last image was this image comes from a scanning electron micrograph. So you can see the three-dimensionality of everything, and you can see that these leukocytes are not just circles, they're actually spheres, and gives you more of a sense of what the blood vessel looks like in relation to the leukocytes that are flowing through it. Here we have a two-dimensional standard electron micrograph just to show an instance in which this leukocyte, which is kind of this darker cell, here's the nucleus and a bunch of other things, probably a lot of ribosomes and things like mitochondria and stuff like that. Anyway, this leukocyte is inside the blood vessel over here, and because of these specialized mediators that come from the damaged tissue stimulating increased vascular permeability, which means basically that you're relaxing the forces that keep the gap junction between endothelial cells tight and sort of close together. Now these endothelial cells are allowed to spread. So here's one over here on the right, kind of looks like the nose of a crocodile or something. And here's another one over here on the left. These two endothelial cells were in contact with each other before this all happened, with maybe just a little tiny space in between. But now this contact is relaxed, and this big old leukocyte is able to shove its way through and between them, so that it moves out of the blood vessel from up here and out over here somewhere where the infection is. This process is referred to as diapedesis, this movement of leukocytes from inside the blood vessel to outside the blood vessel between endothelial cells. It's called diapedesis. Let's see. Oh, this is just to remind me that although I keep talking about a single cell at a time, here's a leukocyte doing this or whatever, the fact is this is happening, it's influencing the behavior of thousands, maybe tens of thousands, maybe even hundreds of thousands of leukocytes all at the same time that come marching through these blood vessels in a parade all the way to wherever the infection is. And so this image here on the right is showing you some infected tissue with all of these spheres. These are all leukocytes that have left the bloodstream, that have moved into the infected area and are now going to do the job that they're designed to do. Okay. Okay, so if we're looking for signs of inflammation, there's some really obvious signs of inflammation that you're probably pretty familiar with already, but I'm going to march through them anyway. The first is that that there's a color change, basically. So if we think about it, if you were to, say, cut your finger, and you weren't real good about washing it up and doing what you should to prevent infection, within even a few hours, but certainly by the next day, you may look at it, this cut that you got, and it's going to have a reddish tint to it. The pathologists have referred to that color as rubor. I think it has something to do with that vegetable. I can't remember the name of it. Anyway, rubor, but to me, it's just red. And also there's a certain amount of erythema that will exist, which will also increase the red color in general. This redness is, I mean, it's no mystery what's going on here. What we've done is we've increased vascular permeability in blood vessels in the infected area, and out come the leukocytes. Well, with the leukocytes come a bunch of other cell types that are much smaller even than the leukocytes, easier to get out of the blood vessel, mainly the erythrocytes, red blood cells. So the red blood cells come out of the vasculature in the area of the infection, and the red blood cells are red. And so this red color that comes out with those red cells creates a reddish color or tint to the skin surrounding the area of our cut finger. So redness, that's probably one of the first signs that something's going on. Second thing that we would notice is that there'd be an elevation in temperature. So we talked a little bit about fever responses before as being a physiological barrier to infection. Well, the fever response is a systemic event. I mean, there are mediators that will move up into the brain and tickle the hypothalamus in just the right way, reset our set point, our temperature set point, and our entire body will increase in temperature. Well, there are also ways in which this increased heat, this increased temperature can occur locally. So surrounding the area, the infected area where we cut our finger, what we would observe is that there would be an increase in body temperature. And if you ever want to do the experiment, the next time you get a cut and get infected or whatever, what you could do is you could take, like if the cut occurred on the index finger of your right hand and you want to do the experiment, you could put your index finger from your left hand halfway up your arm, distal to from the infected area, and make a mental note of what the temperature feels like, and then move it down to where the infection is occurring. And what you'll notice is that there's a significant warmth to that infected area that's not occurring in uninfected areas. So it's just a straightforward demonstration that we can get an elevation in body temperature. And again, it's connected with this physiological requirement for most pathogens to live in an area of normal body temperature. So that elevated temperature in that infected area is really a good thing. It's keeping the pathogen unhappy. Next, what we would notice is that there'd be swelling or edema. This is also no real mystery as to how that all happens. There are fluids and cells that are moving out of the vasculature in the area of the infection into tissues that are not designed to have extra cells and fluids. And so as those things move out of the bloodstream into the surrounding areas, it will kind of push these tissues back, to make space for themselves. And before you know it, you have an area filled with fluid that is really obvious, swelling. So edema. So we have redness, heat, swelling, and then if the swelling continues and the cells and fluids begin to press on some of the peripheral nerves, what we end up then doing is triggering off pain responses. And there could be participation by various mediators like the bradykinins and so forth, but some of it is just a physical aggravation of peripheral nerves in the area of the infection. So we have redness, heat, swelling, pain. Redness, heat, swelling, pain. And so what I normally do to make my point about what the signs of inflammation are is to ask you tonight when you're going to sleep to please chant redness, heat, swelling, pain, redness, heat, swelling, pain, redness, heat, swelling, pain, and then we'll all be on the same page. Okay, thank you. All right, so let's talk a little bit about the cells that are involved in these inflammatory responses. So we have two main kinds that we want to bring up. There's more than two, but we're going to talk about two. One is a collection of cells, actually several subfamilies of cells or lineages of cells. We call them the granulocytes. Granulocytes were named granulocytes because when you look at them under the microscope, they look rather granule. Their cytoplasm is all kind of lumpy, like there's little granules in it. So granulocytes, there are several different kinds of those. Here's a granulocyte right here. It happens to be a neutrophil. And the way I know it's a neutrophil is that it's been stained with a neutral pH dye, and which gave it its original name, neutrophil, neutral pH. This particular granulocyte type, neutrophils, make up about 75% of white blood cells. So 75% of nucleated cells in the blood are neutrophils. Now, as histologists got more sophisticated, they decided that they wanted to name this neutrophil according to a particular characteristic that's associated with it that are common to some of the other granulocytes, not the neutrophil, but other granulocytes like basophils and eosinophils and so forth. So they called them polymorphonuclear leukocytes. And they call them that because if you look at the nucleus, it's this sort of dark area inside the cell. So here's the neutrophil. The kind of lighter grayish area is the cytoplasm, and you can see all the little granules that makes it a granulocyte. And then you look at this darker area, that's the nucleus. Now, when I was a kid, first learning about biology and cells and stuff like that, probably in middle school or whenever, my teacher would ask me to draw a cell, and what I had learned was that you draw a big circle, and then you draw another circle inside of it, and then you draw some hot dog things around, in the cytoplasm. So the outer membrane, and then you'd have the nuclear membrane, circle inside of a circle, and then like mitochondria, ribosomes, and stuff like that. But then when they started examining these neutrophils, they realized that the nuclei they're anything but round. Look at it. It's actually, the way they describe it is lobed. So this neutrophil has multiple lobes. Here's one, 3, 4, 5, I don't know, 6, 7, maybe 7 lobes. It's all the nucleus. It's all attached to each other, and it's all the nucleus. And so that was one thing that was really striking about these neutrophils. But then what they also found was that if you took, say, 100 neutrophils and lined them all up on a microscope slide and then examined them with a microscope, one after another, you'd find that each and every one of them pretty much had different shapes to their nuclei. Some of them, this one has 7 lobes. It kind of looks like a string of sausages or something. Some of them may only have 5 lobes, 3 lobes. They may have 10 lobes. They may be kind of twisted around together in different shapes. In any event, if you look at all 100 of them, you'll find that the majority of them have unique shapes to their nuclei. And so they refer to these cells as polymorphonuclear. Poly meaning many, morpho shaped, nuclei. So polymorphonuclear leukocytes. Nobody wants to say polymorphonuclear too many times in a row, so a lot of people will call them PMNs. Some people will call them polys or polymorphs. You'll call them whatever everybody else calls them in whatever clinic or whatever that you're working in. So these polymorphonuclear leukocytes, these PMNs, most of them, the vast majority, are neutrophils, as I said, that make up 75% of the white blood cells. But the basophils and eosinophils and so forth, they're also polymorphonuclear leukocytes. Okay, so this is one of the main cells that provide us with inflammatory defenses. And here's the second main cell that provides us with defenses, and it is the monocyte. Now, monocytes stain with a different set of dyes, so you can distinguish them pretty easily. But what you can also see is that they don't have multiple lobes. Now, sometimes they do have this sort of, it almost looks like 2 lobes, but nobody ever really refers to them as lobes. They just refer to it as having a cleaved nucleus, this little indentation. And in fact, unless you're a real expert at identifying these cells, if the cell is like spun around away from you and you can't see the cleaved part of the nucleus, you wouldn't even know for sure that it was a monocyte. But the pros know. That's all that matters. So we have this cleaved nucleus. This cell also lives in the blood right alongside the neutrophils and the other PMNs. It makes up about 3 to 5% of white cells. So remember, the neutrophils make up about 75% in round numbers. Monocytes make up about 3 to 5%. Now, the interesting thing about monocytes is that they're not developmentally stagnant. That is, they can change into another cell type. Neutrophils, for the most part, are neutrophils. Once they're developed, they come out of the stem cell pools and turn into neutrophils, and they're neutrophils. But when it comes to monocytes, if they're given particular signals, like during an infection, they can be triggered into a developmental process. We refer to it as differentiation, which the geneticists use to describe cells that will turn off certain set of genes in them that make them what they are currently, and then turn on a second set of genes in that same cell to turn that cell into a different cell type with different functions and so forth. Well, this is one of those cells that can differentiate from one cell type to another. So this is a monocyte, but during an infection, that monocyte can be signaled to differentiate into the famous macrophage. So macrophages clearly have a different morphology than the monocyte have. Also, monocytes tend to circulate a lot. They like to stay in the bloodstream right alongside those neutrophils, but macrophages like to crawl into particular tissues. So if there's an infection going on in a particular tissue, macrophages the monocytes will usually go into those tissues and then will receive signals that will drive them into differentiation so that they become macrophages. Now, monocytes and macrophages will forever be related to each other, and so we often think of them as having sort of a precursor product or precursor progeny sort of relationship. And that's why in some books they'll even say monocyte / macrophage, because they know that those two cells are, they come from the same place. So these macrophages are in these tissues, and they have to perform certain functions right along with neutrophils, in fact, to help slow down the infection. Where do these inflammatory cells come from originally? I mentioned stem cells. I did want to say just a couple more words about that. The process of generating these granulocytes, whether they be macrophages or neutrophils or what have you, they come from a pool, a collection of identical cell types called stem cells, hematopoietic stem cells that, for the most part, in adults anyway, live in bone marrow. So here's my schematic of a bone. It's not very clever, but anyway, it's a bone, and inside the bone is bone marrow, and there's all kinds of stromal cells and all other kinds of cell types. But in there are a population of cells that have a particular marker on their surface that we call CD34. CD34. The CD34 marker, the CD34 molecule, is an excellent way to identify these hematopoietic stem cells. In fact, not only is CD34 good for identifying them, but also monitoring their activities and diagnosing certain scenarios where stem cells are being damaged. And also, we can even use CD34 to identify and then gather up, collect the stem cells into an enriched population that's very useful for certain forms of transplantation. So CD34 is a marker, a molecule that you should keep your eye on. Anyway, these stem cells have two major properties. One is that they divide like crazy. I mean, they make more and more and more of themselves all the time. They're super important, so we have to have plenty of them around. So they're always dividing. And if they are ever damaged, then we're in big trouble. So stem cells have to keep dividing to provide us with more stem cells. And so, whoops, went the wrong way, sorry. So one of the things that stem cells will do, as I said, is to make more stem cells. But the other thing that they're able to do is receive signals in the marrow from various cell types that are, there's too much of it to really discuss right now, but they provide certain mediators, factors, signals to these stem cells that will stimulate them to become other cell types, to differentiate. Just like we talked about monocytes differentiating into macrophages, well, these stem cells are able to differentiate in a most amazing way. They can actually generate every single cell type in the blood from a single cell type. So these stem cells can generate populations of megakaryocytes that eventually will differentiate into platelets, the little bags of clotting factors that are so important that circulate in the bloodstream, or they might be driven down the erythroid cellular lineage to produce erythrocytes, our red blood cells, or down the lymphoid pathway to give us our lymphocytes, or down the myeloid pathway, giving us the monocytes and neutrophils that we've talked about. So these monocytes and stem cell that platelets, erythrocytes, and lymphocytes come from. And these are generally the cells that participate in our first line of cellular against infection. Okay, so we mentioned where they came from. Now the question is, what do they do once they're created? One of their main functions is to perform an activity that we refer to as phagocytosis. Phagocytosis is, in a nutshell, is the ingestion and digestion of foreign particles of any kind, and certainly infectious microorganisms qualify as particles. So phagocytosis is a really important way to clear out, push back on infections as they develop. So first we have the ingestion aspect of this activity, where we have, here's an example. This is an electron micrograph that's showing a bacterial cell. That's this little oval sort of thing here. And it's come into contact with a phagocytic cell. Now this is just a crescent of that cell. It's not the whole cell itself. The whole cell would just circle around and be off the screen. You wouldn't even be able to see the whole thing. So when this bacterial cell comes into contact with the outer membrane of this phagocytic cell, it will attach to the surface of that cell for various reasons, and then and trigger off a response by the phagocytic cell, which results in the membrane, the outer membrane of the phagocytic cell, coming up and around this bacterial cell. And eventually the outer membrane will come up and all the way around the bacterial cell. And when the outer membrane of the phagocytic cell contacts itself, when both sides are coming up around on the other side, you'll end up with this bacterial cell in a vesicle, basically, that we call a phagosome. What's kind of amazing about all this is that this is a bacterial cell. This is a really small little particle. That's nothing. I mean, that's easy to digest. But here we have a phagocytic cell. It's probably a macrophage, because you can see that it's not polymorphonuclear. And what it has done is that it has ingested a yeast cell. Here's a yeast cell. And it is huge compared to the size of the phagocytic cell. Sometimes particles are ingested that are half the size, half the volume of the phagocytic cell. I've even seen some pictures where the ingested particle, when it will push the nucleus of the phagocytic cell right out of the phagocyte. This is not a very selective process, by the way. Basically, these phagocytic cells are just looking for something that is not quite right. It's not natural. It's not self, but it doesn't really know what it is. It doesn't know if it's yeast or bacteria or viral or whatever. It's not very selective. Now, when we talk about the adaptive immune system, you're going to see some real selectivity, but this is not real selective. It's just that frog sitting on a lily pad, and a fly comes by. It doesn't care if it's a horse fly or a dragonfly. It doesn't care what kind of fly it is. It's just grabbing it because it doesn't look right and bringing it inside. Okay, so once it's inside the phagocytic cell, inside this macrophage, sorry about the quality of this image. The contrast is not very good here, but whatever, you'll get the idea, I think. Here we have one of these phagocytic cells right here. I'm circling it. And if you look really carefully, you can see the outer membrane of that cell. And inside this big black blob is the nucleus of the cell. And then all these little dots are parasites or some sort of parasite that the cell has ingested. And within the first few minutes, it doesn't seem like much is going on there, and that parasite probably doesn't really care too much about whether it's been ingested or not. It's just going to sit inside there. But a few more minutes after that, maybe up to 10 minutes later, suddenly all those particles just disappear. They disappear. And it's because they've been digested right down to the basic building blocks of, proteins and polysaccharides and all of that other stuff. So within just a few minutes, they are gone, and you're left with this phagocytic cell that now has been cleared of the parasite. So this is phagocytosis, ingestion followed by digestion. Okay, now we're going to take one little side trip before we move away from inflammatory responses, and we have to talk about the complement system a little bit. The complement system is very complex. There's probably a whole chapter devoted to the complement system in whatever textbook you're using. It's a gigantic pathway of reactions with many, 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 many components. It drives students crazy, drives me crazy, to tell you the truth. But it is an extremely important system. I would like to give you the benefit of my ability to boil it down into just a few essential elements so that we don't have to talk about the whole pathway and make each other crazy over it. Okay, the complement system is a family, really, of blood proteins. Many of them that have a cascading proenzyme-like activity, meaning that they activate each other and promote immune defenses. Let's see. Okay, so in talking about this, what I want to do is sort of give you a prototypic complement scheme. It isn't like this exactly. When you look at your textbook and you say, oh, wait a minute, Krolik told me this, and it's not exactly the same, don't worry about that. I'm intentionally boiling this down to something that's simple enough for me even to understand. So let me tell you a little bit about the complement researchers, the complement biologists. They don't like to go to a lot of trouble naming things. So they tend to name things simply in the order that they're discovered. And so what they did when they discovered the very first complement component, they said, okay, C for complement, 1 for the first one. So C1. And then when they discovered the second complement component, they named it C2, and then C3 and C4 and so on. The trouble is that years later, we discovered that the order in which they were discovered is not exactly the same as the order in which they're actually used in the pathway. We only learned that years and years after they were originally discovered. So there's going to be a little bit of confusion about that when you start studying this stuff in detail. I just wanted to lay that out for you. So let's start with the basic premise that these complement components are, for the most part, inactive. They're circulating in the bloodstream in an inactive state. They have the potential for becoming enzymatically active, able to do various biochemical and biological things if they were to be activated. So we have inactive, sometimes we call them zymogens. And enzymologists will call them proenzymes, meaning that they're not enzymes yet, they're proenzymes. Something has to happen to them in order to activate them. So these proenzymes, when they're activated, and in the beginning, the first complement component can be activated by a number of different things that are connected with infectious processes. So if that first complement component is activated, what will happen is that causes its activation is that proenzyme will be cleaved biochemically into 2 fragments. That's what activates them. So we have 2 fragments. We have the yellow fragment up here and the blue fragment down here. And then the complement biologists that didn't want to go to too much trouble naming any of these things will call one fragment C1A and the other one C1B to indicate that they're 2 different fragments coming from C1. Okay. Then when they started looking at these a little more carefully, they discovered that they actually have different biological activities. So the A fragment, for instance, C1A, might do certain things that have to do with our defenses against infection that we won't talk about right now. But the other fragment, C1B, has one very important activity associated with it. And that is an activity that allows it to cleave and thereby activate the next component of complement, C2. So now this B fragment goes over and cleaves C2, and C2 then ends up becoming two fragments, C2A and C2B, and then one of those fragments is able to cleave and activate C3, and C3 does the same thing to C4 and 5 and 6 and all the way up to about C9. And so there are many steps that are involved, leading to many different complement fragments that can do all kinds of different things to destroy microbial pathogens. But you can see that this is a cascading pathway of activation. And that's why they call it a cascading pathway of proenzymes activating each other, leading to the full activation of the complement pathway and giving us many different opportunities to help defend ourselves against infection. So this is the main premise on how, or the main framework in which complement performs. Remember that this is really, this is just the essence of it. There is so much detail, and it really is very interesting, but it is so detailed it makes a lot of people just a little crazy. At least that makes me crazy. So the complement system, as I said, cascading proenzymes that activate each other and promote immune defenses. They're of the fragments, and there are probably 50 fragments that are generated altogether when all is said and done. I don't want you memorizing 50 fragments, but I do want you to remember a couple of fragments for me. Really, really important fragments. The first one that I want you to remember is C3b. C3b is one of the two fragments that are generated when C3 is activated. Okay. When C3 is activated, there's a C3a that's formed and there's a C3b that's formed. C3b has an extreme important role to play, extremely important role to play in inflammatory responses. It acts as a potent opsonin and performs opsonization. Opsonization is the coating of particles with certain factors like C3b that increases the susceptibility of binding and internalization by phagocytes. Often C3b will stick to the outer layers of bacterial capsules and so forth, so that they can be handled more effectively by phagocytic cells when they come along. So complement factor C3b is maybe the most important opsonin in the body. Now, there are others. One other that we'll be mentioning later on are antibodies. Antibodies themselves can serve as really potent opsonins as well. But C3b, potent opsonization. Remember that. The second complement component that I want you to remember is C5a. Now, it has some very close cousins, C3a and C4a, that perform very similar functions as C5a. But C5a is the star when it comes to these A fragments. C5a can do two major, really super important things. First of all, it is an extremely potent chemoattractant. It attracts inflammatory cells, leukocytes. Now, we mentioned chemokines before as being chemoattractants of leukocytes. They perform very important functions too. They're often released by either damaged host tissue during an inflammatory event, maybe released by endothelial cells of the vasculature. There are a lot of different places that chemokines come from. But C5a is not a chemokine. C5a is a complement component. Please keep that distinct in your mind. C5a is a complement component with intense ability to attract and activate inflammatory cells. C5a also does one other thing down here. So here's C5a, chemoattractant. C5a is also a potent stimulant of increased vascular permeability. Remember, we mentioned increased vascular permeability is one requirement of the inflammatory response. It loosens up the gap junction between endothelial cells in the blood vessel so that leukocytes can wiggle their way through and get out of the blood vessel to the site of the infection. Well, C5a will stimulate that relaxed gap junction. That'll stimulate increased vascular permeability. So C5a not only is attracting leukocytes, but opens the door and lets them out. So what a great couple of things for this complement component to do. In fact, there are others as well that are very interesting, but we don't have time to discuss them at this point. C5a, chemoattractant, and stimulus of increased. And then finally, the complement components, C6, it's actually C5b, through C9, together as a complex, form pores in membranes. They basically will wedge their way into the lipid bilayer of any nearby membrane and create a channel that anything can go through without any resistance whatsoever. So if you have a bacterial cell and you have C6 through 9 forming this channel and in the membrane of the bacterial cell, anything that wants to flow through that channel can flow through it. So we have a lot of the internal cytoplasmic stuff in the bacterial cell kind of leaking out. That's not good for the bacterial cell. But more important even than that is that water, which is very carefully regulated in terms of how it goes into a cell and how it comes out and all of that. Suddenly there is this gush, this flood, a tsunami of water that floods into the bacterial cell. So after a while, like overfilling a water balloon, that bacterial cell will pop because of the pressure caused by the water that's flowing inside. So C3b will enhance phagocytosis of that bacterial cell. C5a will help leukocytes get out of the bloodstream in order to interact with that bacterial cell. And then C6 through 9 will form what they call the membrane attack complex, which is a pore-forming complex that allows water to rush in and other things that shouldn't leak out to leak out. That membrane attack complex often has fondly been described as mediating the MAC attack. Now that just makes me hungry, but anyway, you'll hear people say that every once in a while. Membrane attack complex, membrane attack complex. Okay, so we've gone through as much as I want to go through having to do with basic innate immunity, innate host defenses. There is so much more that I could have talked about, but we just don't have time. And you're welcome to do as much reading as you want, you know, if you want to learn more about these things. It's really cool. Now, the one thing about inflammation and so forth, people don't get too impressed with the sophistication of all of these activities because they're just kind of these big old cells lumbering around, ingesting and digesting things, and so what? Well, the so what of it is that it happens within the first, or starts happening within the first few hours of an infection and for the first few days during the infection before the really big guns of the adaptive immune system that we'll talk about in the next part of this series. It kind of holds things together while the adaptive immune system is coming online. If we didn't have these innate immune responses or innate forms of host defense available to us, we'd be dead before antibodies could even be produced, which, you know, it takes a week for antibodies to show up or any other kind of lymphocyte responses. So without innate immunity, we would be goners. So we have to give proper credit to these cells and these other functions, even though they're not all that fancy, glamorous, and so forth. Really important to our survival. But as I'm hinting, sometimes, in fact, quite often, inflammatory responses are just not enough. Inflammation is just not enough. It will hold things together temporarily, but what we need is for something with much more potent force, much more selective, specific against particular microbial pathogens to come in and save us. And so that's what the next part in this series is going to begin discussing, aspects of adaptive immunity. And I'll see you on the other side.